Hello, everyone. My name is Arthi.
My name is Shan.
My name is Sara.
I'm Jessica.
And we're Team 1 for the 17th Mission Terps in Space competition. We prepared this short clip explaining our intended project idea for this year's M17 mini conference. So the primary goal for our project is to determine the effect of microgravity on telomerase activity. So telomerase is a protein that's located within the nucleus of a cell, and it's tasked with protecting the genetic material in chromosomes by adding non-functional genetic material to the end so that there's no loss of actual functional DNA during cellular replication and DNA replication.
And so the reason that we're doing this experiment is due to the telomerase and its role in telomere extension is because it's involved in many conditions such as aging and cancer. With long-term space travel, it's becoming more and more important to better understand how microgravity affects telomerase activity and telomere length so that future astronauts can be better protected from any harmful side effects that they may experience from extended space missions.
As you can see in the experimental design diagram, our FME will consist of 3 volumes. Volume 1 of our FME will contain freeze-dried yeast. Volume 2 will contain yeast peptone dextrose or YPD broth. And volume 3 will contain 10% formalin fixative. At the start of the experiment, or A0, clamp A will be taken off the FME and will be gently shaken for 95 seconds. And then 2 days later, or A2, clamp B will be taken off and the FME will be again shaken gently for 95 seconds.
So when clamp A is taken off, the freeze-dried yeast will mix with the YPD broth and rehydrate, and then the yeast will undergo growth during which the DNA will replicate, which gives the telomerase an opportunity to be active and perform its function of telomere extension. And then when clamp B is removed after 2 days, the formalin fixative will mix with the yeast, preventing further replication and stopping the experiment. And then when the experiment gets back to Earth, the length of the telomere extensions will be analyzed along with the amount of telomerase mRNA. And then in addition, the experiment will be refrigerated before arriving at the ISS and post-flight.
For our experimental analysis, we will use quantitative reverse transcription polymerase chain reaction, or qRT-PCR, and the telomerase repeat amplification protocol, or TRAP, to analyze telomerase activity of the yeast cells. Telomerase genes TLC1 and EST2P will be amplified to quantify the amount of telomerase, with GAPDH serving as the internal control. The genes that we chose, TLC1 and EST2P, were used since all active telomerase enzymes require these proteins to function.
The expression levels of telomerase will be quantified using green fluorescence. Using TRAP, the size of yeast telomere extension products can be measured, and then we will use polyacromide gel and UV light for visualization, and image J will be used for image analysis.
The size of the ladder on the gel will be suggestive of the telomerase activity, with a larger ladder indicating a longer telomere length and increased telomerase activity. The ratio of telomeric repeated telomerase expression levels compared to the control will indicate the efficiency of telomerase in space.
So conclusively, the results from this project have the potential to assist in long-term space travel by contributing to the understanding of how telomerase functions in space and how drugs and therapies can be manufactured in congruence to the results of this project and ensure that there's normal functionality of telomerase in space.
And so this concludes our presentation for the Terps in Space mini conference. We'd like to give a special thanks to our mentor, Daniel Serrano, and the Terps in Space administrative team for this opportunity and the provided resources. Thank you everyone for listening.